everybody, and good afternoon. I'd like to welcome you all to the next edition of our PFF Disease Education webinar series. Today we have a research topic. This is really exciting and interesting, I think, and we're titling it Of Mice and Men. And we'll be talking about preclinical research, and then as far as the mice topic, and then of men, the PFF's community registry, and you. I have with me today two of my colleagues from the Pulmonary Fibrosis Foundation, Dr. Joyce Lee and Dr. Joe Lasky. And I'm, like I said, just very excited that we'll be having this conversation today about these topics. First up will be Dr. Lee. She is the director of the ILD program at the University of Colorado, and she is also a senior medical advisor at the Pulmonary Fibrosis Foundation. Before I turn the microphone over to her and pop my camera off, I just want to remind everybody, just a housekeeping, if you haven't been on our GoToWebinars before, if you look at the control panel on the right of your screen, there are little triangles for some drop-down menus. If you go to questions, you can actually put in some questions as we go, things that come to mind that you might want to get our group here to answer at the end of our talks. There is a chat drop-down. If you have any technical issues or anything like that, you can drop that down and send us a chat. We'll try to address anything with you there. And then if you are interested in the slides for this talk today, the handouts tab will drop down and you can download those in a PDF format. And to be just a reminder, if you don't already know, this will be recorded and put on our YouTube channel website later on. So you can always go back and review it later. So without any further ado, I will turn the microphone over to Dr. Lee. Welcome and thank you so much for being here.
Thank you, Dr. Case, for this invitation to talk about preclinical studies and drug development. I will do my very best to make this accessible. We are going to be reviewing some data, so we can go to the next slide, Amy, thanks. So this is a disclaimer. Note this is for presentation purposes only. You can read through this disclaimer in more detail in the handouts, but always talk to your provider about any specific medical condition. Next slide. So the purpose of my talk is really to talk about the role of preclinical studies in drug development. I'm going to walk you through two recent examples. One will go more in depth, and then one will be kind of more of a 10,000-foot view, and then we'll talk about what happens after these preclinical studies are done. Next slide. So preclinical studies are really a critical component of the drug discovery process. It's a way to identify potential candidate drugs. And these can either be novel molecules or they can be drugs that we've used for other purposes in the past, and they may have effects on the biology of a disease of interest. So there's a couple different ways that we can identify these potential candidate drugs. These candidate drugs are then tested in lab models of disease to see if there is the potential impact of this drug on the pathways or the biology that's associated with the disease. And this is done in numerous methods, but most commonly it's done in animal and non-animal models of disease. Next slide. Candidate drugs are then tested to identify potential toxicities, how the drug might be processed by the body, and depending on the results of these studies, candidate drugs can then be pushed into testing in humans. Next slide. So every day or every week or maybe even every month, we see lots of headlines in terms of potential therapeutics and in these preclinical studies. And these are really, very common to see, but we're going to go into what they actually are describing to us and what the next steps would be if they were ever to make it into the clinic. Next slide. So the first paper that we're going to discuss is this one that Dr. Noble and his colleagues published in the Journal of Clinical Investigation back in 2022. And this is really looking at the role of zinc in idiopathic pulmonary fibrosis. Next slide. So the rationale for this study was based on the following. So idiopathic pulmonary fibrosis, one of our common forms of interstitial lung disease, is a disease of the aging population. And we know that multiple changes can occur on a cellular level with aging, including metabolic changes. And when we're talking about metabolic changes, it really has to do with chemical reactions in a cell that either produce energy or certain molecules or proteins. And what is known is that zinc is an essential element that maintains some of these metabolic functions. And low zinc levels and regulation of zinc has been associated with response to injury in animal models of disease. So that was the premise for this study. Next slide. So these investigators looked at several different things. And so I'll kind of walk through the methods for this study, and then we'll take some time to understand the data that they shared in their publication. So they looked at the expression of genes in certain cell types of patients with idiopathic pulmonary fibrosis. They looked at what's called alveolar epithelial type 2 cells, which is a lung cell, and they compared them to cells in patients without IPF. Then the next step they did was to identify a gene involved in zinc metabolism. And then their next step was to determine the role of this gene on certain biologic pathways associated with fibrosis. Next slide. Finally, they investigated the role of this gene in mouse models of pulmonary fibrosis, and they then went on to look at the role of what we call correcting this deficiency in the gene in the mouse models of pulmonary fibrosis. Next slide. So their first step was to identify a target of interest. And so what they found was that there was this zinc transporter called ZIP8, which is a protein. And this protein was decreased in the cells of patients with IPF, so in tissue, lung tissue cells of patients with IPF. And then they identified that this zinc transporter protein is controlled by a gene called SLC39A8. So the protein is regulated by this gene, and then they looked at studies of this gene in patients with IPF. And so what you can see in the figures below, we'll go left to right, and these aren't all of their figures. There wouldn't be enough time for going through all of them, but I picked some key ones to show you. So in the one that's labeled F, they are looking at the expression of the gene that controls the zinc transporter protein. And in those special lung cells of the lung called the alveolar epithelial types 2 cells, so AEC2, the gene is expressed lowly compared to healthy lung tissue samples. Then in panels H and I, they looked at other data sets to look at gene expression of this SLC39A8, found similar findings in not only other published data sets of IPF, but also organoids, which are little tissue samples of patients who have IPF. When we go into panels K and L, you can see in IPF patients in K, their percentage of cells that have this protein called ZIP8 is lower than in healthy controls. And then similarly, the gene that expresses or regulates the production of ZIP8 is also lower in IPF patients compared to controls. And then the final one that I show on the right is the relationship between DLCO, so remember that's the diffusion capacity for carbon monoxide, and the relationship with the expression of this gene. And so patients with lower DLCO had lower expression of this gene. Next slide. So their target of interest really has to do with these things. So the gene that they identified was SLC39A8, and this gene regulates the production of the protein called ZIP8. Next slide. So the next set of findings that they reported in their manuscript is really trying to determine the role of this particular target in the lung. And so what they identified was that ZIP8, this protein, transports zinc. So they found that there were lower levels of zinc in these type 2 cells of the lung compared to healthy controls. They also discovered that ZIP8 and its role in zinc metabolism is needed to renew this cell type in the lung. So these cells can regenerate in the lung, and so they identified that this protein was critical for the renewal of these cells in the lung. And then finally, they found that zinc affected another pathway associated with fibrosis called SIRT1, and that this relationship is dependent on this protein. So these panels down below also walk through those findings. You can see in panel B that the number of zinc positive cells in patients with IPF is lower again compared to healthy controls. The panels F and G demonstrate that the ability of these cells to regenerate or renew are dependent on the presence of zinc. So in panel F, both in healthy controls and IPF, you can increase this colony forming efficiency, or CFE, by administering zinc to these tissue cells. In panel G, you can see that if they take out the ZIP8 protein, that they are unable to create these cells in the lungs. But if they have the ZIP8 protein, that they're able to augment this renewal of the cell type. So really, these are all steps that were taken to try and understand the role of zinc biology in IPF patients and how they compare to controls. And finally, the right-hand panel really demonstrates the relationship between this other fibrosis pathway called SIRT1, zinc, and the protein ZIP8. Next slide. So here, the results of this kind of set of studies is looking at the role of the ZIP8 protein in the lung. So what they identified is that it does have a response to lung injury. It affects zinc transport into the cells. And then it is also involved with another important fibrosis pathway. Next slide. So here, then they transition. So all of the studies that I've shared with you so far were really done in cells and specific cell types. And so the next step was really to look in animal models. And the most common animal model that we use in pulmonary fibrosis is that of the mouse model. And they did 3 different mouse models of disease to try and understand the role of zinc and ZIP8 in these animal models. So the first one is looking at aged mice. So looking at older mice, they found that there was less expression of the ZIP8 protein in older mouse lungs. And the older mice had less of a response to zinc treatment compared to young mice. The second model really injures the mice and tries to create scarring and inflammation in the lungs, and thought to represent to a certain extent what we see in humans. We know that they're not identical models, but it is the model that many therapeutic agents are tested against. So in this injury model, they found that there was impaired recovery of the injury in old mouse lungs compared to young, that mice that were deficient in zinc had a worse survival and worse lung fibrosis after injury. You can see in panel G. And then those mice that were given a higher zinc diet had decreased lung fibrosis after injury. The final mouse model that they created was really to take out ZIP8. So they created a line of mice that did not have this ZIP8 gene in it. And these mice who lacked the ZIP8 protein developed spontaneous lung fibrosis when they were older. And in those old mice, if they were given this injury with bleomycin, had worse lung fibrosis. Next slide. So in their studies looking at the role of their targets, so zinc metabolism in mouse models of pulmonary fibrosis, they found that normal zinc metabolism and zinc supplementation decreased response to lung injury in mice. And zinc deficiency, whether they did it by nutritional deficiency, aging of the lung, or removal of the gene, increased the response to lung injury in mice. Next slide. So what this study tells us that in mouse models of disease, zinc regulation impacts the mouse's response to lung injury and the development of fibrosis, and that zinc regulation may be an important modulator of lung fibrosis in humans. Next slide. What this study does not tell us is the role of zinc supplementation in patients with IPF. So clinical trials are still needed to determine whether or not zinc supplementation is beneficial in patients with IPF, but also whether or not there's any risk associated with that supplementation. Furthermore, this study does not tell us to prescribe zinc supplements to patients with IPF. Next slide. So that was the zinc study, and then I'm going to share with you kind of some high-level findings related to saracatinib and updates regarding what the status of this particular drug is in drug development. Next slide. So in this study, they used methods to look at existing drugs and to see if they could be what we call repurposed based on mechanisms known in IPF. And with their study, they identified a potential drug called saracatinib. And then they directly investigated this saracatinib in 3 preclinical models. And they compared the preclinical models to nintedanib and pirfenidone. And the preclinical models that they used included human tissue cells, mouse models of pulmonary fibrosis, and then human and mouse lung tissue samples. Next slide. So I mentioned that this was this is based on a repurposing or repurposed drug. So saracatinib is a dual kinase inhibitor, and it was an experimental medication originally developed by AstraZeneca for the treatment of cancer. So this medication was never moved through to FDA approval because it was stopped in phase 2 due to lack of efficacy, but they are looking at this medication in a different light to see if it might be relevant for patients with pulmonary fibrosis. Next slide. So here are their primary findings. So after they identified saracatinib as a potential drug of interest, they found that saracatinib blocked certain fibrotic pathways associated with pulmonary fibrosis. Then they found that saracatinib altered gene expression associated with pulmonary fibrosis. Saracatinib then reduced the fibrotic and inflammatory pathways in those preclinical models. So again, these are in human tissue cells and in mouse models. And then the findings that they found related to saracatinib was similar or better than that observed with pirfenidone or nintedanib in the preclinical models. Next slide. So as a result of this study, the investigators have moved saracatinib into a phase 1 and phase 2 study. So the clinical trials number is listed there. And their study is called saracatinib in the treatment of idiopathic pulmonary fibrosis, or STOP-IPF. In this study, they're giving once daily dosing of saracatinib for 24 weeks in 49 participants. And the primary outcome measures of this phase 1 and 2 study is really looking at safety, tolerability, pharmacokinetics or pharmacodynamics, which is how the body metabolizes the medication, and then the change in forced vital capacity over 24 weeks. Next slide. So we see a lot of headlines regarding preclinical studies, but it is a long process in order to get to an approved therapy for the treatment of any condition. And so what we've focused here today on is really this first step here. So this in vitro and in vivo testing that's really centered on basic research and early drug discovery in these preclinical studies that we've just reviewed here today. This process can take anywhere from 2 to 5 years, and candidate drugs in the very beginning are maybe 5,000 to 20,000 candidate drugs. Those that pass like preclinical standards, really it reduces to maybe 5 or even 10% of these potential agents that really demonstrate efficacy from a preclinical standpoint. Once preclinical studies show some promise, then investigators can choose to take them into what we call human testing, so the classic phase 1, phase 2, and phase 3 clinical trials. And as many of you know, this process is very long. It can take 3 to 7 years to move a potential target of interest across these different clinical phases. And through that process, only 5 really make it to through the each phase of this process. And then finally, once a drug goes through a phase 3 process and shows efficacy, it has to be reviewed by the FDA, which can take up to a year. And then only one of these agents end up getting approved. So you can see kind of the funnel that is created as a result of this process. So there's a lot of promising preclinical studies, but at the end of the day, what gets approved for use in patients is much smaller. Next slide. So I think from my standpoint, what to take away from preclinical studies is to know that progress is being made in the understanding of the biology of pulmonary fibrosis, and that's truly critical in order to identify novel drug targets. And these novel drug targets and repurposed drugs are being identified by many investigators around the world. Next slide. And while the majority of these drug targets that are identified in these preclinical studies will not survive the gauntlet of human studies, some will, and there are, as many of you know, many ongoing and upcoming phase 3 clinical trials. So as a physician who cares for patients living with pulmonary fibrosis, I encourage all my patients to be encouraged by these findings. People are still working very hard to identify better therapies for patients living with this disease. But these are really preclinical studies, and we don't recommend taking any of these medications or supplements described in the preclinical studies. And if you have questions about that, you should really discuss them directly with your doctor. Next slide. And I think that's it for me, and happy to take questions from the audience when the time is right.
Thank you so much. Yeah, so just as a reminder, while I get our slide deck switched over, if you have, oops, I don't know if you guys can see that or not. If you have questions, you can enter them in the question tab on your control panel. And let me get our next slides up and welcome Dr. Lasky back to the screen and share these with you. Sorry, give me just a minute. All right, hopefully you can see that. My control panel tells me that you can. So here we have, all right, great. So this we're going to talk a little bit now about the Pulmonary Fibrosis Foundation's registry projects. And as I mentioned before, Dr. Joe Lasky is joining me for this. He is our Chief Medical Officer at the Pulmonary Fibrosis Foundation. So he's a great person to have this conversation with us. And I'll also bring up, if you didn't already know, that April this month, we are really talking about and encouraging people in our PFF community to join our PFF community registry. So we wanted to highlight this during this month's webinar and encourage everybody here, if you haven't already done so, to be a part of this important research as we go forward. So thank you, Dr. Lasky, for joining me. And I'm gonna stay on camera this time, and we're gonna have a little conversation about the registry. We say this a lot. I know you see a lot of communications about the registry from the PFF. Dr. Lasky, can you talk a little bit about let's take a step back and say, what is a registry and why is it important? Because it's obviously different from the preclinical studies that we talked about.
Right. Thanks, Amy. I'm very excited to be able to convey and encourage everyone to get involved in this registry. We're talking specifically about the community registry, which kicked off in around July of 2022, and we have over 1,400 patients involved in the registry, but we're trying to shoot for 500 between this month and next month, additional folks, and I hope we can make 2,000 by the end of a year of this up and rolling. And so the registry serves and helps us serve the community of people with pulmonary fibrosis. That's the purpose of it. And it's a way of having a dialogue with patients who have pulmonary fibrosis to understand how the disease is impacting them. And we're able to collect various data depending upon what the registry is. Now with the community registry, we're not able to obtain blood samples and things such as that, but we are able to obtain how the disease is impacting the patients. In this case, not only patients with pulmonary fibrosis, but those who've had lung transplants in the past, and those family members who are caring for patients who have pulmonary fibrosis. So with this information that's gathered in a very careful and specific way, in a very data safe way for this information, you're using guidelines that are provided by the national government, we're able to collect and collate this information and be able to query it to get answers to the questions that people have. And we have many questions right now that a registry could serve like this. Well, we have other questions that we haven't even thought of yet, but we will in a year or two, and this data will be available to be probed at that time to answer those questions. So again, in keeping with the original purpose of the Pulmonary Fibrosis Foundation as a patient advocacy group, this helps us do that.
Okay, that's a wonderful description. I think this, it's just really to think about the contrast between this and a clinical trial, for example. This is really just a collection of relevant, important information, and then the ability to query that, to ask questions of it, to try to answer questions of it at a later date. So we'll talk a little bit more about that. This is going to give us an opportunity to talk about the difference between our patient registry, which has been going on for several years and has collected a lot of data, versus the community registry moving forward. Joe, do you want to talk a little bit about sort of those differences? You alluded to the ability to collect, blood samples and maybe a little bit more clinical data in one way, but not the other.
Right, and this is a registry that was started, the concept was 8 years ago and actually kicked off about 6 years ago and finally closed after it had run for 6 years of time. And this is something that Kevin Flaherty was very involved in, but he wasn't available today to walk us through this registry. But this registry was conducted at the clinical sites for the Pulmonary Fibrosis Foundation. 25 of the sites collected the data on these 2,000 patients and then followed them for as long as they could for a period of 6 years. And we're able to collect some CAT scans and some demographic information and also how the disease was impacting the patients as time went on, as well as some blood samples that could be queried later in order to try to understand pulmonary fibrosis better. So that was that registry. And now moving into the community registry. So the community registry has some advantages, and I already mentioned that we're going to be able to talk to caregivers and lung transplant recipients as well as people with pulmonary fibrosis. And it doesn't matter what caused your pulmonary fibrosis, we're interested in knowing about it. And because it's not centered on these care centers, then it's more widely applicable that anyone in the United States can log on and spend the time informing us about what their experience is. So it broadens, who we can reach. It doesn't limit it to those who could go to one of these care centers.
Absolutely. I think it's so important. The sky's really the limit with the ability to include members of our community in this registry and touch on issues that we may not pick up on in people who have either proximity or the resources to get to a care center in person and get their care there. So I think that this is such a great tool and to be able to build this information. Also want to just mention, I think this sort of leads into our next slide, but the data that was collected in the initial phase of the patient registry, it still exists and it's still there to be utilized by researchers. And there are lots of questions still to be asked of it. But I like to show this here as just a small sample of the information that's been obtained and the studies that have been done and published from that initial registry of 2,000 patients, and what came of the information that those patients donated to these efforts and their participation and time. So Joe, can you talk just a little bit more about that and kind of the importance of it?
Sure. Well, you know, there's 50 either publications or abstracts already published from the registry, with a dozen more that are ongoing and will be published within the next year, and a few dozen more that we haven't even thought of yet. That this data is available, will always be there, as well as the biospecimens that are available to answer these questions. So, you know, this has been very impactful, and it is something that we never could have done without the participation of patients signing up to be in the registry. So that's why we're making this appeal right now for the community registry as well.
Absolutely. I think it's just so fascinating to see how our community, how the researchers in our community have utilized this information, how the centers came together to recruit patients to participate, how the patients came together to donate their time and their information, and how the researchers have now gone in and utilized all of that to help us understand better associations, just the kinds of things that you can understand from a registry. You can't necessarily test a new therapy in something like this, but you can look at associations. You can generate additional hypotheses. You can understand impact and of disease and you know, other factors on patients that are living with pulmonary fibrosis. And those things have really informed how we understand and our approach going forward. So I think it's such a valuable tool, and we value the participation of our community on all levels. So I said just now that we can't necessarily test a new therapy in a registry, but Joe, could you talk a little bit about the precision study? Because it is a testing of a therapy, and it connects to the registry itself.
Right, Amy, this is something that when the registry was first started 6 years ago, that this wasn't even on the radar. This is something that cropped up. And so there are things that are happening, you know, because of the registry that we didn't dream of when it was being conceived of. And so too, I think this will be the case with the community registry, as we talk about that a little bit more as well. But this precision study is a partnership between the National Heart, Lung, and Blood Institute that was never possible without the registry being present, in which we're able to look at a specific genotype of patients who might respond because we're not all the same genetic code to a certain therapy better than others. And this time it's N-acetylcysteine, or NAC. And we're 3/4 done enrolling. We expect to be finished enrollment with this by the end of this calendar year. And we're very excited about this type of trial that we were able to do, which is called it's precisions because it's precision medicine. We're not all the same, and we're not all the same genetically in our exposures and such. And so we're trying to understand how some therapies work better in some people than others in regard to this. So this is very exciting and the first of its kind, and could not have been done without the registry.
Really incredible. Okay, so another sort of spin-off, I guess, is kind of the way to think about it, or something that's working in concert with the registry. It's the Prolific Consortium. Dr. Lasky, I have a little trouble getting my head around this. Can you explain what this is and why it's important and connected to the PFF's registry?
So with this prolific consortium, we got all these drug companies that are sitting on your screen here to interact with the Pulmonary Fibrosis Foundation because they view us as an honest broker in this regard. And we had the biospecimens, and we had what happened to patients over time. And so working with these companies, what we're looking for is something called a biomarker. Now, biomarkers can be used to determine maybe someone has a disease, but they're not symptomatic yet, and so you want to find it early. That's one type of a biomarker. Another type of a biomarker is you look at the level of whatever's being expressed in the blood, and here we're looking at 12 different proteins in the blood, and saying, do these correlate with what's going to happen in the future? Will somebody get better more quickly or more slow, or have less of a decline over time, depending upon the level of these biomarkers or combination thereof. And this may even help future studies. If we identify biomarkers that can predict very rapidly a changing in the biomarker, whether a drug's going to be effective or not, it can accelerate the movement of drugs from these concepts that Dr. Lee was talking about with the mice to actually your doctor can write a prescription for it because it's approved by the FDA. So it's all an effort to accelerate diagnosis and treatment options.
Fantastic. All right, so actually have a couple of questions already typed in about what you can do. And since we're talking about joining the community registry, this is one of those things. Dr. Lasky, do you want to walk through these points and just mention to people how they can get involved and help?
Well, this registry is not hard to find right now because we're making the push here in April. It's front and center on the website. As you go to the pulmonaryfibrosis.org website, this is what will pop up, and all the questions you could possibly have, frequently asked questions, what it's about, and what kind of a time commitment is involved. You know, the time commitment might be an hour twice a year in order to do this, and that's an hour you can break up into 15-minute segments or 20-minute segments, do from your home computer in order to log this in. So there's no travel involved or anything like that, or expense involved as well. So that's what you can do as listed here. Amy, do you have further things to add?
Well, I was going to say exactly what you were saying. It's very easy to find at this point because it is on the front page of our website. There is a process that takes a little bit of time to get through. I will also add that if for any reason you run into any technical issues, we do have support. You can email us, and we do have folks who can get back in touch with you and help if there are any, you're running into any login issues or trouble with getting into the system or something like that. So we've got plenty of help. If you just please don't get discouraged, let us know if that comes up, because sometimes the tech is not always perfect, and so we want to make sure that we've got support in place for everyone to be able to participate who wishes to do so. And then I would also just remind everyone, I know we have patients who have logged in, but this is open to patients, family members of patients, caregivers who are not direct relatives, spouses and so on, who are involved, and as well as folks who have had a lung transplant for pulmonary fibrosis or interstitial lung disease. So this is a broad invitation to join and help us answer some of those questions and some of those that we haven't even thought about. So this is a great way to do it and get involved. And so again, just to remind you that we're looking to get over the next 2 months or so up by 500. We're knocking on that door, and so hopefully we do get there soon. And if you're interested, just don't log off early because we'll answer some questions here. But when you do, you can go there and get involved. So you've got if you want to take your phone, you can actually screen this, do a little shot on this QR code here if you know how to do that, diddly boo kind of thing.
Another thing I'd like to emphasize, and sorry if I cut in too early, I wanted to catch in there. One of the things that this registry allows us to do, which we didn't do in the site registry, was to start a conversation back and forth with their patients. So if people have other questions that maybe we didn't put in the questions that are in the registry, let's say, just for an example, we may have questions about, are you involved in a clinical trial, and why not, and what are the stumbling blocks, and try to get that information. And with that information, we can pass it on to the drug companies that are, and the NHLBI that are doing the clinical trials, to try to facilitate and make it more easy for folks to get involved in clinical trials and participate. Because the reality is only about 5%, 1 in 20, is getting involved in a clinical trial. And through this type of a registry, we may be able to get that conversation going so that we can make it easier, know what the stumbling blocks are, make it easier for people to get involved in clinical trials and help us find the solution. So that's just one example. Amy and I came up with 2 or 3 others earlier today of where we may want to query you and have you give us the information. Well, we don't want this information from just a small focus group of 10 people. There's error in doing that. We want thousands of people to tell us, so we get a very good answer that's well thought out from many different perspectives as to what needs to be done.
Absolutely. Yeah, it's I think it's really important, and like you said, there are so many ways that this could be used in the future. So please come and join us. I'm actually going to go back and leave this slide up while we talk, and we'll invite Joyce or Dr. Lee, if you'd like to pop back on with us. We'll try and answer some of these questions that we have from our audience today. We've got some great ones, and I may just kind of go back and forth. Hopefully our heads don't spin too badly, but go back and forth between topics so that we can share the microphone a little bit. I'm going to go to Dr. Lasky first. This is a question about the registry. If you were in the patient registry before, are you automatically in the community registry now?
No, you're not automatically in the registry. You would need to sign up, and we encourage you to sign up.
Absolutely, yeah. So that original project has stopped collecting data now, and effectively participation in that is ended, although the data that's collected is still, it still exists and is still being used. So consider this sort of a new phase of the project, would need a new sign up and consent for the community registry. Oh, it's a very good question. So technical question about some of the preclinical things, and Dr. Lee, I'm going to make it twofold. One is, why do we study mice so often rather than other animals? Dr. Lasky, feel free to jump in on this one as well. And how does one create, you mentioned creating a mouse with a missing protein or an altered gene. Not to get too technical, I mean, you can be, I think you can be sort of basic on this, but how does that work? Do you just, where do you get those?
Those are both great questions, and I am certainly not a bench scientist, so I'm grateful to have Dr. Lasky here on the call with us as well. I think mouse models have been used probably for a variety of different reasons, convenience being one of them. The size of the animal makes it a little bit easier in the laboratory to study than larger animals, but I do know that some investigators do larger animal studies, not just mouse models of disease. But that, I don't know, has historically been the animal in which many of the drug targets have been tested in. I don't know, Dr. Lasky, if you have other comments in terms of why mice became the primary animal of choice here in the bench world.
Well, there's many reasons. One of them is it's cheaper to work with mice than larger animals. That's one. Another one is that the technology for doing this started in mice some decades ago, and it's become easier and easier. Now most universities have a specific service that an investigator such as us here, we could say, we want to knock this gene out, or we want to change one protein within this gene, and then be able to inject that into the mouse and have a mouse that expresses a certain protein or a mutated protein in a specific cell type. So it's really gotten technical over time. But the nice part about it is that there are services within the universities that took what used to take us 6 months to do, weeks in order to get at least the injections done in regard to these mice. Mice aren't the perfect model. Mice aren't people, and with every model, there's pros and cons of doing it. And I think more and more lately, we've decided that we need to do our studies in old mice, because IPF is a disease of older people, and cells don't react. Old cells don't react like young cells. So this is you can imagine having to raise a large colony of some other animal till it got old in order to do these studies. So mice are still a good model, and we're learning a lot from mice, but it's not the end all of models. And I do want to say that the NHLBI has put out competitions for proposals, and are funding 5 or 6 different proposals with different types of models that don't use mice, that use lung slices from the lung of people, or other mechanisms forming organoids from cells and such. So we're trying to refine these models as we move forward.
That's a great answer. Thank you, guys. Okay, so back to follow-up question, Dr. Lasky. What do you mean by old mouse?
Yeah, well,
it's a good question, right? Like, it makes sense.
Yeah. So a mouse only lives about 2 years, and so if we get mice up above a year or up to a year and a half, then we call that an older mouse.
Makes sense. And it seems like throughput would be a lot easier, since, as Dr. Lee pointed out, these preclinical studies, the number of things that are being looked at can be in the thousands or even higher. And so something that could be, move more quickly and be high throughput makes a lot of sense for trying to make an impact. So back to the registry, a question here. Can a person who's participating in a different registry, let's say at their care center, they have given permission to collect their data, maybe they're giving blood samples, they had a lung biopsy, something like that, or can they also be in the community registry?
The short answer is yes.
Excellent. I think the short answer should suffice. There's really, I think longer answer is if you're collecting data and not intervening, then there shouldn't be necessarily anything wrong with giving data to both sources. We wouldn't want somebody necessarily to be participating in 2 clinical trials that are testing novel medications, for example, because those things, may not, shouldn't overlap like that. But the registry shouldn't be a barrier. Okay, I have another question here. Sorry, I'm just rolling through some of these. This is a really good question. So maybe on other websites, forums, things like that, I've heard about the Zinc study and some other studies about supplements or something like that. Now, Dr. Lee, you mentioned that the Zinc study doesn't show that patients with pulmonary fibrosis should be taking zinc, and we shouldn't be advising them to, or at least doctors shouldn't be advising them to do so. But would it be okay just to get some from the store and start taking it? And could there be a downside?
I mean, I think the things that are still unknown about zinc in patients with IPF is how much zinc does somebody need to take in order to have a benefit. And so while zinc perhaps in a multivitamin or something like that might be okay, we don't know how much zinc would be needed in order to achieve the effect that they're seeing in the mouse models of disease. And so how much zinc to take would be certainly a question. And then as you take more of this supplement, there will be negative consequences to taking additional zinc, and what those are and how that might impact a patient living with pulmonary fibrosis. I don't think we know the answers to that as well. So I would strongly caution against just taking extra supplements just because you think it might help, because we don't know the potential consequences if you take too much or not enough. I think there's a lot of unknowns when it comes to things like zinc supplementation.
Absolutely. And I think just a follow-up question on that, there are, as you mentioned, repurposed medications or therapies that are being looked at and have maybe shown some promise in preclinical trials. Would you say similar things for other things like, I mean, NAC is being studied, or N-acetylcysteine is being studied in precisions, but or something like metformin, which has been, that's been a little newsy over the past couple of years. You would make a similar recommendation for those types of things? Is that true?
Well, yes, because metformin, as we know, the clinical indication for that right now is for the treatment of diabetes. And if you don't have diabetes and you start taking metformin, you could have problems with your blood sugar levels. And so I do think that we don't know the effect of these repurposed medications without the clinical trial that tells us whether or not we should use it. An example that comes to mind is our study on Coumadin. So we looked at Coumadin in patients with IPF, not for the purpose of anticoagulation or thinning of the blood related to atrial fibrillation or stroke or other indications, but really looking at that as therapeutic medications for patients with IPF. And that study was stopped early because of increased harm. So I think that just is a study that demonstrates that while there might be preclinical studies that show efficacy of an existing FDA-approved medication, until we test it in the patient population and look at how it affects the disease of interest, it's really hard to recommend any of these preclinical agents in the treatment of our patients.
Absolutely. So I have a couple of questions about specific agents, but I'm going to use this rather than address those specific agents that have been studied in preclinical trials to suggest that if something has come across your radar and you're interested in knowing, are there human studies going on that, and how close might that be to inhuman, or to use? What phase might it be in if it is? One option would be to go to the Pulmonary Fibrosis Foundation's clinical trial finder. That is, you can find that linked from our website, and it brings together clinical trial information for things that are ongoing right now in humans. And so if there's something out there that you're thinking, I wonder if this saracatinib, for example, is, I saw the preclinical information, I wonder if there's a human study at this point. We know now, because Dr. Lee told us that there is, we should be able to go there and look and see, is it there? It's at phase 1 and 2 right now. Where are the sites? Is that something that's accessible to me if I'm interested? And there is also an opportunity to reach out about that study to sites to help on our clinical trial finder. So if you're interested in getting involved in a clinical trial, we talked a lot about the registry, but in a clinical trial that's looking at novel therapies, the clinical trial finder is a great tool to be able to see what is out there, what piques your interest, what might you be a fit for, because there are certainly inclusion and exclusion criteria for all of these studies, and how is it accessible? Where is it? And might you become involved in an ability to reach out for that? Dr. Lasky, any thoughts or comments on that? I know this is something you're very interested in, is clinical trial recruitment and the ability to do so through the trial finder.
Right. There's been some wonderful improvements on the PFF website. That's very laudable, and you've been very involved with Amy, so you know much about it. The other thing about this community registry is, there is the option to, can we contact you if there is a clinical trial near you? Maybe there's a clinical trial that's very early and it's only being done in Schenectady, New York, and of course, we want to get this information as quickly as possible, and they only want 20 people, and so we might be able to, we can now with this registry, if you click the box that says we can do this, we can say, okay, people within a 2-hour of Schenectady who meet this criteria, this trial's going on, and would you be willing to help out, and the trial may not be a medicine, a trial might be something like, is there a new way to determine more quickly whether the medicine works? Maybe some imaging, a PET CT scan or something like that. So there's a lot of good work that can be done through the communication that's going on through this community registry.
Absolutely. Dr. Lasky, I'm going to send another one your way, and that is, why do we see so many clinical trials that are not more inclusive? They're looking primarily for patients with idiopathic pulmonary fibrosis, that specific diagnosis, when many of our patients don't have IPF, they have other forms of ILD. Why are so many of those studies looking at just IPF? And will we see more options? Are we seeing more options for more broad types of interstitial lung disease?
Very, very good question. And there are options for people with progressive pulmonary fibrosis out there right now that are not just limited to IPF, but the person who asked the question is right. The majority of them are focused on idiopathic pulmonary fibrosis. I think there's probably a lot of reasons for that. I think we have a better understanding of what idiopathic pulmonary fibrosis is going to do over time, so we can design the clinical trials in such a way as to know how many patients we need to meet to reach a certain endpoint that's been approved by the FDA for drug approval. That's one example. I imagine Dr. Lee and yourself, Amy, could think of a few more reasons.
Yeah, I think that definition, sort of the clean definition of IPF and uniformity, always seems to help when you're designing a clinical trial and trying to predict or hypothesize what the outcome will be. But I'm very encouraged. I have had those same questions myself, and for my practice and for my patients, I'm encouraged that we're seeing more investment in patients that don't have idiopathic pulmonary fibrosis. I mean, still investments there, but other patients that have particularly progressive pulmonary fibrosis, and those who have other types of interstitial lung disease that are less fibrotic, that there's interest in finding new and better therapies for those patients as well. Okay, we are almost to time, so I am going to, even though we have more questions, I'm sorry, I am going to forward through here. We have answered our questions. We are going to thank our sponsors today, those who have contributed so that we can have these webinars. And then I'm going to just thank again my colleagues, Dr. Lasky and Dr. Lee. I really, really appreciate you being here, participating today. This has been such a useful and interesting topic of conversation with lots of engagement and questions. Finally, I will remind everybody, or tell you if you haven't heard this before, if you are, when we log off of the webinar, there will be an opportunity immediately following will pop up to give us some feedback about our webinar and hopefully help us plan better for the future. So if you would please stay, fill that out, give us a little bit of feedback. There will not be another opportunity to come to you later. And also to remind you, if there's anything you missed, this will be recorded and posted in the coming days. So thank you all for being here today. Really appreciate your participation and engagement, and we'll see you next time. Have a great afternoon.
Bye, everyone. Thank you.
Thanks for attending.